We're going to talk about DNA replication. So all living things are made of cells. For growth and repair of organisms, these cells have to divide to make more cells. Here in our drawing, we have a fertilized egg, which is the sperm and the egg already together. We call that a zygote. And then that cell needs to divide to make an actual fetus, and then from there you make a baby. So the first cleavage will result in 2 cells, and then the second will result in 4, and then the third will result in 8, and so on and so on. And during this time, we have more and more cells being made. So the process of cellular division is very important because it makes every cell in an organism genetically identical. So we want that DNA to be exactly the same. So the process of dividing into 2 cells is only a small part of the cell cycle. Most of the cell's time is spent preparing to divide during interphase. So during interphase, like we read in our reading, we have G1 phase, S phase, and G2. Then we have mitosis, which is broken down into 4 different phases or stages. And then we have cytokinesis, which is at the end where the cell splits in 2. And 90% of the cell cycle will be happening in interphase. So for cellular division, the most important thing that happens during interphase is the synthesis or S phase. It is during this stage that DNA replication occurs. So here we have a drawing or diagram of DNA replication that is happening. So every cell needs a complete set of DNA in order to be considered an actual cell. And this means that before the cell can divide, DNA must be replicated or copied to make 2 complete sets during the S phase. So here, during DNA replication, here it shows us a parent cell with 2 different chromosomes. And then after DNA replication, we'll have the 2 different sets because it was copied. So before we can have DNA replicated, it must unwind and the 2 strands must separate between the nitrogen bases. Here it shows us 2 different drawings of that happening. And you can see here the previous strand is being separated and copied. So a really important enzyme that is used is the enzyme DNA helicase, and it breaks the hydrogen bonds that hold the nitrogen bases together. So we have it here represented by a blue triangle, and it'll come in and break those hydrogen bonds between the bases, and then either side will be copied. We call this unzipping the double helix because it's literally kind of looks like a zipper and it's unzipping it. So the place where DNA unzips is called the replication fork. And here we have a drawing of that replication fork. Let me go back really quick. And this is what we see when the DNA is unwinding and unzipping. And then it will have a copy of a new strand being made on each side. So after we have our helicase unzip our old strand of DNA, another enzyme comes in, DNA polymerase, and they add nucleotides to create two complete and identical sets of DNA. So here we have helicase separating the two, and then we have DNA polymerase here represented by some rectangles, and that is going to put all of these nucleotides together so that we have two new strands. Let me make sure you guys wrote this down. Okay, so as DNA polymerase adds these new nucleotides, so on one side we have our old strand of DNA, and then we have DNA polymerase adding the nucleotides to make the new copy. Another second polymerase will proofread the process to make sure everything is correct on the DNA strand. So here we have it, we have the new things coming in, new nucleotides, and then we have like a double check just to make sure. So remember Chargaff's rule, adenine A and thymine T always go together, and then guanine G and cytosine C always go together. So DNA replication is called semi-conservative process. And like we were talking about during the reading, we do not get one old set of DNA and one new set. So the reason it's called semi-conservative is because we really have one old strand paired up with a new strand, and then the other set will have an old strand set up with a new strand. And that's how DNA replication works. Let me go back and make sure you guys wrote this down. So we call the old strand the parent strand, and then the new strand the daughter strand. So here in our picture, we have our old strand coming from our old DNA, our double helix, and here it's represented in white. And then the new strand, which was put together by the different enzymes, DNA polymerase, is in blue. So again, we have the old strand pairing up with a new strand, and then the other old strand pairing up with another new strand. So there is our parent strand, and then we have our daughter strand. So go ahead and write down how many chromosomes we have in an original cell. So if in the original cell we have 46 chromosomes before the S stage, how many chromosomes will be in the cell after the S stage? So if you remember, during the S stage we have DNA replication, or when DNA is copied. So if we have a copy of 46 chromosomes, how many chromosomes in total will we have? If you wrote down 92, you are correct. It's just 46 times 2. So since the S stage results in 2 complete sets of DNA, there would be 92 chromosomes after replication. So go ahead and write this down. Okay, so that is the end of our notes. To make sure to have everything that was in red and green written out. If you didn't get something, make sure to get it from any of your neighbors. And we will go over them in class tomorrow a little bit more in depth. But make sure you have everything written down. Okay,